So at any rate, the lecture. This is okay. So there is, what is that? That's a mouse. Okay. And here's another mouse here. So this is somewhat infamous. It's called my mouse-to-mouse lecture. So we're going to start with a mouse. We're going to end with a mouse. And there's a lot of mice that aren't going to enjoy everything in between. But before we get there, I want to at least make sure, kind of touch base on what I think is one of the hardest things to sort of understand about how antibodies were made. And the reason is this: if you think about it, you have DNA, and if there's one thing we kind of try to do as organisms is kind of like maintain our DNA. So then, if we're trying to maintain our DNA, then how do we make antibodies to an antigen that we're exposed to? And then end up making those antibodies to defend ourselves, because it means that you have to have the ability of your DNA to make millions of antibodies. In fact, they've estimated that the human body, based on the system, can make up to 10 billion different antibody epitopes. And that can recognize a lot of different things. And so that's the idea, is your antibodies, there's a system that's set up there, and I'll attempt to make teach you immunology in 5 minutes, okay, how it works, okay. So we'll start with that, and then we'll erase it because it's not really part of the mouse-to-mouse, but the product we're after is antibodies, okay. So, and why are we so interested in antibodies? Well, one reason would be what?
Because they help defend yourself.
Yeah, come on, something better than that. Thank you. Money, okay. Okay, so it's estimated there's about $200 billion in the pipeline for antibodies for therapeutics and drug companies right now. And that's a lot of billions. Okay. So 200 billion, I think that's the number that I remember correctly. Quote me if I'm wrong, but I don't know. It's a lot, a very, very lot. Meaning there's an awful lot of drug companies that are going to live or die on the pipeline of antibodies that they're developing. And the magic of antibodies is they bind very specifically to things. Extremely specifically. So specific, we haven't found anything better than this. Okay. And then how does it actually work? Okay, pardon my analogy, but first, okay, there's B cells. And B cells are highly uncommitted, let's call it that way. So it's kind of like college guys, okay? So these are or like anybody in college, they're kind of not sure what they want to do when they grow up, okay? And so in the process of doing this, there's a really cool mechanism. For those of you that think you really understand enough molecular biology, go and try to read the wiki page on antibodies, because now it's good. Compared to what it was a few years ago, it's really hard. But the basic concept here is that what an antibody is it's this thing that looks like a Y. It doesn't really, but okay, it kind of has a thing on the bottom here, which is the constant domain, and it has the variable domains up here, and it looks more like this, like that as an antibody. And what it does is these regions out here, there's 10 billion ways that it can modify that to stick to something. And if it sticks to something, it usually means it's bad, it wants to kill it. And so how does it generate all of those? Well, it turns out it's a really nifty thing. It has these domains that are variable, that are tandem all beside each other, and then it has another series of a bunch of other ones that are determining factors, and then it has another ones here of some other factors that are here. And it randomly goes through and it cuts out like this one here, and then this one and this one get together with that, and it decides out that this one and this one will go. And all of this ultimately results in it putting together a DNA sequence that it finally grows up and becomes committed. Sometimes that commitment is good, and sometimes that commitment is bad. How does it know if it's good or bad? Well, depends if it finds a mate. Okay, so you commit first, then you find second. Okay, so they actually decide on making the antibody, and these things, the B cells, then they'll cover their surface with 50,000 or so antibodies, 50,000 antibodies, and so it's kind of like deciding what you want to do when you grow up, and then you hope there's a job. Okay, so you have all your capabilities, your affinities, whatever it is here, and then you wait for the thing to come along. That is this, is that perfect mate that happens to bind to that with reasonably high affinity. Now, that process then from there isn't sort of quite enough, because a B cell needs to then mature from a B cell into a serum cell. And a serum, this is one that basically decides. So that this thing undergoes, if it binds to something, then it gets to multiply. You can see my analogy. Okay. But it multiplies, and in the process of multiplying, it also has another mechanism that is, introduces single DNA point mutations into this thing. And so as it's amplifying, it's also then taking that variable region here, and it's changing it just base pair by base pair, which of course makes a slightly different antibody. The one that binds better then interacts with another thing in your immune system called T cells. And the T cells feed back, and if you're the lucky one, if you say that like this one here makes Y, and this next one here makes Y prime, and this one here makes Y prime prime prime, or whatever, and this one binds better, then it gets to make more copies of itself. Okay? The one that doesn't bind as well actually undergoes the process of cell death. Anyone know what that word is in biology?
Apoptosis.
Apoptosis. Okay, so that's something, a word that 10 years ago never would have heard. Certainly no chemie would have. Okay, and so they get to die. Okay, and these guys here. And so there's this really cool innate process that your cells, by the way, knowing where these were. Where were the B cells? Here they were. Here's the B cells. Where do you find B cells? Okay, so it's basically in two big places, are your spleen and your bone marrow. Okay, for people like me who lost a spleen when I was 10, falling out of a tree, okay, I have to rely highly on my bone marrow. Okay, so in theory, I'm immune compromised. But now I'm particularly, I could use as the example of this thing is like just before I left for India, I had to take live typhoid. Okay, and so they gave me little pills that had live typhoid in it, and then I needed to develop an immunity to typhoid in case I went swimming in places like the sea off of Mumbai, which I did. And so it's going to have to bind to that thing. And this is a pretty big thing that it's trying to bind to. It's not like talking about a DNA sequence. And so as I'm doing that, hopefully one of the gazillion B cells in my body decided to bind reasonably well to some epitope on the surface of typhoid. And then in the process of doing that, went to the rest of my immune system and said, you do that. And in fact, it's kind of like, well, marriage would be a good word if it actually was commitment for life. Okay, but it is like that. So in other words, once they do that, you get to live on for quite a long period of time, and then those cells now, oh shoot, you're supposed to get a booster after, so I'm supposed to go get another round of typhoid pills that I take 6 months later, just to, in case these guys were going good, to jazz them up one more, and then I'm good for life. I can go and drink typhoid. Okay. So that's how the thing works, and it's kind of important to understand this because it's going to be related to the whole process we're going to talk about next. If you don't get all of that, you can just say that, well, you inject something with an antigen, it makes antibodies, and then you try to isolate that and make money. Okay? So there's immunology 101. Okay. So let's say now we want to make antibodies to do a diagnostic test, because that's where a lot of it came from. The example I used to use all the time was like, for instance, HIV. HIV positive means they do an assay that shows that you've been exposed to it. You don't have symptoms yet, but you have been exposed, right? Let's use something less scary. Chickenpox. So once you've been exposed to chickenpox, then let's say that I wanted to make an antibody that I could use to find out whether or not you've been exposed to chickenpox. Okay, since they no longer have the chickenpox parties when your kid got chickenpox, and you invite all the neighborhood kids over, and then they all play, and they all get chickenpox at the same time, and that way one or two moms can then watch the chickenpoxy kids. Now most of you probably had vaccine, right? Yes, no, a couple of hands. How many of you have been vaccinated? So some of you have been vaccinated by the Merck vaccine for chickenpox. I hope it works. Okay. If not, it's really kind of nasty to get chickenpox when you're older, especially if you're a guy. Okay. Keep that in mind. So I could take the chickenpox, and I would want it to be the surface, so I might take, I don't have to necessarily inject chickenpox into said mouse, but I could just get the capsid protein, for instance. I could even express it in E. coli, hoping it would fold right, etc. But this is sort of the starting point for how we make antibodies. So we introduce antibody making 101. It is inject antigen into mouse. Okay. If I want to scale up, how do I scale up?
You get a mouse to call for the mouse.
What's that?
You get a mouse to call for the mouse.
No. This is an immune response. Its spleen somewhere in here is going ahead and thinking, I want to make antibodies to this thing. And so I have to inject a certain amount of antigen relative to the weight in order to get the correct immune response, to get that. Okay. Well, what happens? How do we get, first of all, how do we get our antibody for our diagnostic? Here's one, huh? Okay, so this is where we start. Okay, so we take the mouse, okay, and we put the mouse in the blender. Okay. So now it turns out that there's where do you find a lot of antibody, which is also kind of cool. Okay, because you can eat something and end up with it the antibodies ending up where? And where the antibody? Where's the highest? If I wanted to get your antibodies, and I was an antibody stealer, where would I do it? No, I don't want your spleen either. I don't have one. Intestines. No. Blood. Okay, so blood, your blood. So you'd have to bleed the mouse. Okay, and then not much blood in the mouse. Okay, so if we were going to scale this up, what would we have to do?
Use a bigger animal.
That would be, see, 2 routes, right? More mice, more injections, more blending, okay? Or we can go to, and this is perfect for this time of year, what would be the next best animal, or one that you might have heard about? It's time appropriate.
Rabbits.
Thank you. You're on the ball today. So unfortunately, of course you can tell my skills here. The Easter Bunny, okay, would be a bigger animal, right? That's one way to scale up. And so you can get rabbit antibodies. In fact, back in the Dark Ages when I was there, rabbits were in fact not an atypical thing for me. If I wanted antibodies doing research on a protein, I could make my protein, send it off somewhere, okay, and I would get it back, and apparently they would find rabbits, okay, because rabbits have more blood. Okay, what's the next one up? Thank you. Okay, you can see you guys are good. No worries. Okay, I say so. That is a rabbit. This is a goat. Okay, so that's one way to scale up, right? Okay, it would be, but obviously there's a lot of downside to this because if you think about it, in addition to making something, okay, there's something else that you have to do to use the antibody you're going to make. What's that? Can I just take rabbit blood and then I start doing research with rabbit blood?
You need to.
What? What?
I'm trying to think.
Okay, trying to think.
Time.
Okay, next. Okay, so let's say that let's say you want typhoid antibodies. I just took a course of typhoid pills, okay? So if you wanted typhoid antibodies, you could bleed me, okay? Then what? Is that the only antibody I have? No, I mean, obviously, in fact, the old-fashioned way, when I went to India 20 years ago, they actually used to, instead, they didn't have all this nifty stuff, is they would actually collect an antibody bolus, put it into a big glycerol thing, and give you an injection in the butt of human IgG antibodies. So they would collect antibodies from a gazillion college students, and then you inject them with it. Now, you kind of, maybe that's not, sounds so good anymore, right? Because the last thing I want is thousands of college kid blood in my behind. That would be a real pain in the, never mind. Okay, so, yeah, thank you. Okay, so, separation is a pain, right? I mean, obviously, any of these, the blender is really bad, right? Because I have all that other protein and stuff. Blood, at least, is very full. There's a lot of serum protein and a lot of antibodies there. But there's going to be a lot of other antibodies, right? In fact, there's a lot of antibodies that are in there that are very generic antibodies. And they are made before something has been exposed to them, so that you have at least some immune response. Because one of the things antibodies can do is just kind of stick to things and therefore make them like not be able to move around and stuff. So they're more like that. So if I really wanted to do this better, what do I want to grow? Do I want to grow rabbits? In the back, what do I want to grow?
Mice.
What's that?
Mice.
Because mice are easier than rabbits. I was going to have a demonstration and set a trap last night in Fensky because there is actually some mice. Okay, but I didn't. No, we don't want this, right? We want something different. We want something in vitro. So what are we going to grow?
Use CHO cells?
CHO cells. It's out here somewhere.
Okay.
Okay. We're a long way away from the other mouse, the human mouse. Okay, so just for a moment, if I could, what would be the obvious thing that I would want to do?
Stick your hand and pull it out.
What? What are you going to pull out?
The antibodies in there.
Okay, no, so close. Okay, I was going to show you my scar, but I won't. Okay. Who said it?
The spleen.
The spleen, right? I mean, in other words, you don't want to grow the whole mouse, okay? It upsets PETA when you drop it in there, okay? And they wouldn't be quite so upset if we could grow spleen cells, for example. In fact, you know, we basically want to grow the cells that make the antibodies, not all the cells. In fact, it would be better yet if I could actually grow just the spleen cell that was committed. Okay, you all know how hard it is to find a committed spleen cell, right? Just not even looking at it. Okay, so let's say we do at least try to do that. So we'll just, you know, we'll go to our rabbit, we'll do surgery. Okay, and so the Easter Bunny now, we'll take the Easter Bunny and we'll try to find, you know, we'll take the spleen out. You can live without spleen, I know that. And try to do that. Now, one of the fundamental problems, okay, so let's say this is your spleen cell. First of all, you'd have to know the one that decided to make the antibodies. That might be hard, but it's a way to do that in a second. But there's something else inherently wrong with this approach. And that is that human cells have a very limited lifespan. Okay. They don't. They only divide so many times. If they're not a stem cell, they're already committed. And then after that, they undergo apoptosis. Why? Because if they didn't, they would be cancer cells, okay? Or they would accumulate mutations that would increase the likelihood of them being a cancer cell, that kind of thing. Okay, and so this thing here, basically, you have, you know, they die. Okay, so eventually, it's a good idea, but it has a limited ability. So the other option then is it turns out that you can actually fuse cells together. So you can actually take the spleen cell then, and instead then take a cancer line, okay? Which cancer, anything with an oma on it, right, is, it says a myeloma, that's kind of close. So you take a cancer cell line and a spleen cell line, and you literally, you put a little polyethylene glycol in there to disrupt their membrane so they go together. And I end up with what's referred to as a hybridoma. Okay, so hybridoma was basically the fusion of the spleen and that. And now you would typically do this. So you do take one mouse, okay, and then you take the mouse and you inject him, and then you actually just then you inject him a couple of times, okay, because you really want to amp up his spleen to make an antigen to your thingy, because you want to increase the probability that there's quite a few spleen cells in there that have figured out how to be committed to making the antigen to whatever you're injecting. Okay. Then you do the fusion with this, and then there's some different ways you can do this. In the most painstaking way you can kind of imagine is that you can do immunochemical screening where you end up with multi-well plates. You've seen these things like this. Okay. And each one of them you dilute it to a single cell in each one of those there. And the simplest way of doing this is you actually take your antigen, you conjugate it with a enzyme that will do a catalytic assay to make a color. Then it's kind of easy. You look for the one that actually has color, and then you're, oh, that's the spleen cell I want, and take it, and then you can do that, and then take the hybridoma line that's been selected, and now you can grow it. Okay, so this is, now how do you grow hybridomas? Well, it depends a little bit. Let me see, there's a guy, he's on the ball here today, so, okay, so there's that, and then here, try. Okay. Okay. Not so on the ball. Okay. Okay. Wasn't trying to throw it to you. It was him. He was looking at me. What you have there is called a T-flask. Okay. It's made out of polystyrene. It's very light. Okay. And it's used a lot. It's very cheap too, and it's easy to gamma radiate, so you can sterilize them. So these things are used by the bazillions. So if you want to grow them, one of the fundamental problems now is you've now figured out how to get to the point to make a cell that's making your product. But it turns out these cells are really hard to grow. There are two basic problems, or two basic challenges, I would say here, is that finding the right kind of media to make them happy is hard, because in this particular case, cells that like to be, remember cells like to be around other cells and so forth. And they're also used to being very catered to. In your body, you have nutrients all brought to the cell, waste all taken away. And so in a sense, it's not like a bacteria that's used to being on its own. These guys have to deal with, with that. And so media was one problem. And the other problem was attachment dependence. Attachment dependence comes along with being committed. Okay. Or not committed, or whatever that. Okay. So the media. So let's say that I wanted to make a human cell or a mammalian cell happy. What would be a media that you might use for that? I already mentioned this sort of the other day. If I was like, again, think about it, if you were just given the problem of, hey, you got these cells in here, they're very unhappy, do whatever you can to make them happy. Let me flip this point. Years and years ago, when they started to do plant tissue culture, they figured out that I could extract sterile cells from a plant, put it into culture, and grow it. But it didn't grow very well just giving it sugar and other stuff like that. So what kinds of things might they have added to plant media? Take a guess, anyone here?
Light.
What?
Light.
No. They're working on sugar. They don't need light. They don't need to do photosynthesis.
Ammonia. Water.
Carrot juice. tomato juice. I mean, in other words, if you wanted them to feel like they're you're going to grow them in tissue culture, then why not add juice? You know, plant juice. Okay, and that would do that. In fact, one of the best medias for plant turned out to be coconut milk. Okay, and so you actually went and we used to harvest pina colada parties. Okay, so by analogy, what are we going to add to our hybridoma to make them happy? This isn't a big step. This is a small step. No? It's on the board. Okay. Okay. Mouse juice. Okay. Now it turns out that it's hard to get a lot of juice from mice because they're kind of small, and so instead we use bigger animals to scale up. Okay. What's that? Cow. Okay. Yeah. Okay. And in fact, you want it to be relatively clean. You don't want to throw the whole thing in there because we need to make hamburgers, right? Okay. But it turns out that you can take the serum, okay, and in fact, it turns out the best serum of all is from fetal, and so this was the so-called FBS, fetal bovine serum. Okay. Yes, baby cow juice. Okay. Okay. Now, we're probably not making Pete any happier now, okay, that we're not throwing mice in the blender, but it was this, and fetal FBS would cost about $500 for 500 mil, like this big, jar that big, okay. But you can kind of imagine it has a lot of good stuff in it. If I'm a cell and I want to grow and hormones and vitamins and all that stuff, a lot of proteins and stuff in there too, right? And so that was one of the media components that were there. Now the other problem is the T-flaps that I sent around. So you guys, where they all end up there. Okay, so they're attachment dependent. Okay, so that T-flaps thing that you see there is, it looks like this. It has a little bit of an up on it in here because literally what you would do is you put a pipette inside there, pipette them inside there, pipette in a little bit of fetal bovine serum, okay, and then they would grow on the bottom until they became confluent. I showed you pictures of that last time. So everybody touches everybody else, and they're all in that. And so that was the way that you could grow them inside that. And in fact, then they would secrete antibodies, and then you could take the antibodies and dump the juice off, and from that isolate your antibodies. Okay. So if I wanted to go from there and I wanted to scale up and I'm an engineer, what do I do? Well, one of the answers, go ahead, hold up the T-flaps. There's one, where's the other? There's a little one, okay? There's a bigger one, okay? So that would be one way to scale up. What's wrong with that type of scale up? It's not a big pipette. I have big pipettes. Fundamentally, the problem with this, it's a geometry problem. What?
As you increase the volume proportionally, there's less surface area per volume.
There just isn't much surface area, period. So let's say that I wanted to do a better job with this, what would I do? Because the way it works, by the way, is you actually take that, you set it inside, pull it up a little higher, into an incubator, and then you maintain pH control by having a 5% CO2 and a little bit of carbonate in there. So pH control is based on the carbonate equilibrium, and it just sits in there static like that. Yeah.
Could you have trays?
Okay. Well, you could, like lots of them, and there's one downstairs in the pilot plant that has lots of trays where you can do lots of experiments on it. But you can kind of see our problem is the amount of surface area you get is pretty small. Now what would be the simplest way that you could say that I'm going to double my surface area? Nope, 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 throw it to me. There, just doubled it. Okay, I just turned it around. I used both sides. That would be one way. And that became the basis of the roller bottle. Okay, now I looked for my polystyrene roller bottles and I couldn't find them all. And so, this is a $200 roller ball. Okay, But you can kind of imagine this thing actually goes on into that and then it just simply rolls. And that's how you grow that. So they would grow on the outside. What if I wanted to increase the mixing and the surface area in something like this? How might I do it? As I spin it faster, that it doesn't get any more, right? They're the surface. What?
Really shake it on the other axis while it's turning?
No, because it's going to coat completely. The whole inside of this thing is going to coat. I was going to bring one, but I didn't thought it'd be. They make them out of polystyrene that it goes like this, right? So there's little hills and valleys. You can imagine that you get another double. But that still isn't a lot of surface area. So this basic concept of increasing this. So here we go to roller bottles, which go around in circles. Okay. You can imagine that it has liquid in here and it's rolling around in circles. And I told you already that that's how they scaled up the original EPO-based process. Rooms like that, rooms the size of Fensky, with roller bottles everywhere you can go, 12 high, that people running around hairnets tending roller bottles. Okay, that's how it was. It's worth a lot, obviously, as a drug. Okay. So what if I wanted more surface area? What could I do? Anyone? What's that?
Show some. We're almost there.
Oh my gosh, there yet. Okay. Engage brain. I want more surface area than that. How can I get more surface area? I know you want to answer. I can see. No? No? There we go. Ria? Rio? Rio. Yep. This is any come on. Just and get I want more surface area in my culturing thingy. How am I gonna get more surface area? What?
Use a few back
So it's going to be, what, like the ice cream ball you kick around? Yeah, I'm trying to, okay, yeah, that's minimize, you took calculus, right? So you're minimizing the surface area for, no. I mean, your answer was almost exactly right, except really bad. Particulates. Little spheres, right? So what if I actually take the thing, and I take it and I do what are called microparticles, okay? So now, okay, I'm going to just fill my reactor full of beads of polystyrene, okay? Now I can get a lot of surface area in here now, right? I mean, okay, so this is bead technology. Well, it's kind of hard, you know, you're going to do this, right? You can still attach, right? But I need to now mix it, right? So now it's starting to look a little more like chemical engineering, right? Tank, but I don't have to be worried about not beating up too hard on the beads. And then, of course, by the way, chemies are never satisfied. That's probably a commitment problem. Okay, no. How do I get more? I mean, I want more than, you know, I solved this. I mean, we've gone the whole way for, oh, I forgot the worst one in between here. Okay. Let me just take a tiny thing here, and let's say what would be the best possible reactor for my hybridomas. Yes. Okay. In fact, okay, the best one of all is the mouse, and so the intermediate, by far, which was much better than these other technologies for a long time, and I'm sad to say that I did do this. Okay. Was to take the hybridoma and you take that cancer line and you inject it into the peritoneal cavity of the mouse. And so the mouse then basically grows a big tumor inside of it. And then you actually harvest, then it, of course, you know, it blows up with a thing called ascites, A-S-C-I-E, I don't know how to spell ascites. Okay, but that's the juice that you blow it up with when you have a big tumor growing in your inside. Okay, and then you blender. Okay, now you suck it back out. You stick a big needle in and you suck all the juice out. Okay, and this was the you could get really high titers here. Because here where you were, think about it, we were waiting for some spleen cell to make a little bit of antibody and so forth here. I select for the one that does that, then I put it back into the mouse, and then the mouse makes an entire organ inside, called the tumor, okay, that is making nothing but the actual antibody. And so, and it's nice and concentrated, lots of protein inside here. This was actually, for a long time, it was hard to beat this. Okay. Where's that? We don't have our vegetarian here today. Okay. This is bad enough, okay, for everyone else. But you can kind of imagine, so you kind of, we're not on PETA's good list yet with that one, right? Because that's what you're trying to do. You're trying to, if you think about this, you're trying to mimic it. Now, back to this, okay? We have a bead technology, but the amount of cells we can put per density is very low relative to the volume of tumor inside of a mouse. So we need more, right? How can we get more surface area per unit volume of our reactor?
Make the beads porous.
Beads porous. Okay, so now you make the beads with, you know, little porous holes in them, right? And so, as you can imagine, at this point, then, in fact, these cells can grow in here, and that might even mean they don't get beat up by the impeller as much, so on and so forth. But for every problem you fix as a gimme, you make another one. What is the problem we're making? Wrestling guy in the back. You don't know. Okay. You don't see any disadvantage to beads full of thingies here. What are we trying to do? It's all about keeping your cells happy, right? We were keeping them happy by putting baby cow juice in there. We were making them happy by putting them in a mouse, okay? Is this going to be happiness for these guys? Guy right there.
No,
Why aren't they going to be happy?
they're not going to be able to get oxygen or anything else. They're going to get stuck in
Good. Okay, so we basically introduced a mass transfer limitation now. Okay, and so as you can imagine, this bead, okay, is now going to have the oxygen and the sugar, H12O6, all diffusing in now, because before it was easy to get to them, right? It was sitting on the surface. Or they're sitting on the surface of the bead, but now they're inside the bead. And of course, they're going to pee in the media, and so all that stuff like the lactate and other products, waste products, are going to have to diffuse back out again. And so you've introduced much more surface area, so then there's different things you can do. So let's say, for example, that I wanted to go one step further, and I've introduced a mass transfer limitation, right? What would be the way in which I could increase the mass transfer? Because I will have both diffusive mass transfer limitation in here, right? And I will also have a boundary layer mass transfer around the bead itself. Well, I can make the holes bigger. I can play certain games there with that. But let's say I wanted to get rid of the boundary layer mass transfer around that. What is the way in which we can decrease the mass transfer limitation around our bead? Her first. You're next.
Make it more porous with smaller pores.
Does it matter? Does the boundary layer change due to a change in porosity on the inside of the bead?
No, but you can make an infinite slab when you don't have the boundary layer.
That would be our T-flask, right? Yep. So yes, but we already decided that we didn't want a T-flask. Okay. We want to use our beads because of the surface area thing, so we want to increase the mass transfer around it. Go to you. That's you.
When stirring,
Stirring to a certain extent will, okay? But remember that they're relatively small, okay? And when you say the stirring, what are you thinking in terms of? What is that stirring doing? Your thought is right, but when you want to improve the boundary layer heat or mass transfer around something, the way to do that is to do what? If I am paying the heating bill in Fensky, which is supposedly 50 grand per month, something like sucking all the air out of the building. So, that just pains me. Sorry. If I wanted to, on what days would I have more heat loss from the building? Say it.
Windy days.
Windy days, okay? Started in this training, don't. In your, okay? You need to increase the relative velocity between your object and the thing around it. Now the problem with stirring inherently is that remember, if I'm going around with the flow, which I kind of have to, because if I'm not going with the flow, then my eddy lengths are going to be small, and they're maybe going to be messing with my cells and making them unhappy. So if I go with the flow, I don't actually make the relative velocity go up that much. I may move around the tank more faster, but the relative velocity that gives rise to the boundary layer for mass transfer may not change very much. So if I want to increase the relative velocity between the particle and the liquid, how can I do that? Make the beads stationary. So we're going to tether the beads. No, that would be one way of doing it, right? Because you could, in fact, take it and now make a packed bed of beads, right? And then just like we did for our high fructose corn syrup, we could increase the flow rate past it, and then we would be able to do that. The problem is that this isn't quite like an immobilized enzyme, and the length of period of time you're going to do that, it's kind of tough to do this, where you increase the flow rate and pressure drop. What would be another way that I could make the relative velocity faster?
Could you just make the beads heavier?
Yes. Okay. And so you get a patent and start a company called Virax. Okay. So Virax said, hey, wait, if we just take our beads, okay, and we put in little, I forget if it was titanium or something like that. I guess it was around the time of Terminators. So little beads in there that are titanium is very heavy, okay, and it's inert, okay. It's used for implants and stuff. It'll sediment much faster, and in fact, it'll sediment so fast. What I can do is the inherent problem with this is the issue of channeling and stuff that you get inside beds. But if you have a fluidized bed, where you actually take the beads and then you have an upflow and go around in circles where it goes up there, the sedimentation rate now will be very fast. The relative, you can actually eliminate the boundary layer mass transfer. And so beads that were porous with little thingies in them to make heavy was another technology that was used to try to overcome this problem of mass transfer limitation. Okay. And in here you didn't transfer much oxygen, so you go back around, and then in here you would actually have a hollow fiber setup. It would actually do that. Oxygen transport. And you can kind of imagine, so you're trying to do this. Now, you can kind of see what we're trying to do here. What we're trying to do is get a higher and higher density of cells in a reactor, kind of like the tumor, right? So why not go all the way to that and try to actually grow essentially a solid mass of the cells themselves? Now, what's the problem with achieving a solid mass of cells? How does the tumor overcome this fundamental problem? Some of you may know this. What are most anti-cancer drugs? Some of the ones for very aggressive cancer, what do they do? Do you know?
Well, then they spit out the drug really fast.
Not you. Let's see if we can get somebody else here on board. In the green. Okay. If a tumor tries to grow really fast, it also is going to run into the same problem of mass transfer limitations. So what does the tumor tend to do? So we're back to our tumor here. Okay. So if I was to look at that tumor inside the mouse in there, what would I find to be a characteristic? How did it get away with growing a big, solid mass of cells? Because obviously that's going to be much more mass transfer limited than the Virax beads or this other stuff, right?
Transfers to other parts of the
See what?
body. It transfers to other
I mean, let's face it, how do we do it? We're pretty big, okay? We're not amoebas, okay? And so how do we get oxygen transfer? What?
Fluid?
Yeah, say it.
Blood?
Blood, circulatory system, right? I mean, in other words, in order to get it there, so what happens is it turns out that tumors they actually promote the form of angiogenesis. The way they feed themselves is they do that. So many anti-cancers actually go after, like Avastin and some of the others, they actually prevent it from making the blood cells to feed itself to try to choke it off. Okay? And so that's the fundamental limitation here. So all we need to do is build a reactor that has a lot of capillaries inside it, which would be called a hollow fiber reactor. Okay? So a hollow fiber reactor, then, it has thousands of little tiny fibers that go through here, from here to there. And then the cells can grow on the outside, and it's kind of like little big blood vessels, right? And you can kind of imagine that this type of technology is going to run into the problem of how big I can make it, but it depends on how much oxygen is being consumed. And if I'm going to be consuming the oxygen here, I'm going to have to put a pair of lungs up here somewhere, right? Okay. We actually put in another hollow fiber reactor with air or oxygen in it. And I put another unit that I can actually take out some of the waste that's coming out of it. And so the original one of these, I actually have one downstairs in the basement of the Fensky, all dusty somewhere. It had a name, and it was called the Micro Mouse. That was the commercial name of the unit. Okay, so because it was like making a big mouse, right? It's a big tissue. It had a lung. It had oxygen exchange. It had a waste product removal thing like it. It was a reactor system where you could continuously perfuse nutrients into it, and then the waste product that would come out of it, waste, and it was full of antibodies. Now these are referred to, by the way, as monoclonal antibodies. I should have said that here, the spleen cell and this whole process here. A polyclonal antibody is when I inject it into the mouse, and then it makes multiple epitopes. And so let's say here's my nasty thingy, right? This is my antigen. And so I have one antibody that sees this part of it, and one antibody that sees that part of it, and one that sees this one over here. It's because as these B cells mature, they'll make to whatever part of it, and they'll get better and better at grabbing onto that. And so a polyclonal antibody means that it is a mixture of antibodies that recognize different sites. Okay. A monoclonal, since I selected a single cell here to do that, I select just one of these guys. And so I've functionally chosen one of them, and therefore I'm making, in all these cases, monoclonal antibodies, and that's MAB. You'll see that a lot in the literature. So, everyone get the micro mouse? Everyone can see the problem with scaling the micro mouse. Is that it's kind of hard to make a hollow fiber reactor, plus if I make it longer, now I'm going to have enough oxygen from one end to the other. Okay, you get some nifty things in here called Starling flows, for instance. As I increase the pressure drop across there, running through the pipe, okay, that'll create a pressure drop. It'll actually cause a little bit of tissue or juice to move through the outer spaces. Okay, and if anyone's ever followed, like the reason why people may lose so much weight in space is because they don't like their bones need to like have that kind of squishiness to get stuff to move around in that. That's why they strap them in and make you do a lot of exercise, so you don't have too much bone loss. Okay, sorry. Okay, okay. So at any rate, so we're headed in the direction, and it turns out that these started to get close to what we could get out of this. So this was kind of the original replacement for the little cancerous mouse, was the hollow fiber reactor micro mouse. Now all of these were still predicated on a very important thing, and that was this issue of the attachment dependence. Okay, having to them to be that. So now what came onto the scene is CHO. Okay. So, and it was right about the right place. Okay, so what we needed was a attachment independent lines. That is, ones that could grow as a free cell suspension. then here, then what you had, and this was like Chinese hamster over cells, CHO cells. And in fact, the genetic engineering of moving the ability to do that was a huge advance. If you wanted to make, now see, because CHO cells don't just have to make monoclonal antibodies, but they can. They figured out how to move the DNA components to that into CHO cells so that that would be that. And CHO, to this day, had remained the standard way of doing that. And now if I want to make more money, what do? I'll go to the back, since I didn't give you a chance yet. If I want to make more money with the same capital investment and everything I've got here, what do I end up doing with my system? I need to scale it up. I don't really want to scale it up. I want to make more money. How do I do that? Two ways. More cells. Perfect. Okay, and so that basic concept where this went eventually was to what was referred to as perfusion culture. For years they said that this was all done in batch. You stick them in there, you grow them up, you harvest it, and that. When they came to perfusion, they just came up with various ways to keep the cells inside the cell, and they would run at very low flow rates through the system and let the cells all come back in here and grow these to pretty high densities. And so this, and many of these runs could run 6 months to a year. And so you could now do a huge amount, because when you go to continuous production from batch, this transition from batch to continuous usually is at least greater than 10-fold increase in productivity. For any process, this isn't just bioprocesses. This is why we do chemical processing in continuous mode. That's why we focus on that so much in Chem E, is because batch doesn't fully utilize the capital investment of all the toys. But you start real low and you go high, and it's the average of that time. If you can operate continuously at high productivity, continuous systems do that. So this became the basis of running lots of CHO cells inside them. So, problem semi-solved. I already mentioned the fact that then the next step, the next problem now, was not really an engineering problem. What is wrong with our antibodies that we're now making here?
They're not natural.
They're not natural, say one more word. Not natural, I'm not sure. What's that?
They're not like naturally occurring.
Well, they are naturally occurring because they came from nature. You're like 9.5. They're not human. Okay. So that's the real problem. We got to the point where you can make lots and lots of antibodies, but when you're using them for diagnostics, way back here with this, and just bleed a few mouths to run some gels and see if you have proteins in that, it's entirely different if I'm going to talk about repeatedly giving a dosage to a human. Because if I do that, remember their immune system is also going to be saying, hey, wait a minute, what are you doing here? You're injecting antibodies into me that are from a hamster? Okay? And so that became the primary issue here. From here was this idea of humanization. Now, I already told you that there is this company called Glycophi, which I'll put these in because you can, when you're bored talking to your parents, it happens about 10 minutes, okay, after Easter dinner, okay. And instead of going and putting on the Bourne trilogy, you can go ahead and put on the, I don't know how to spell any of this stuff, by the way, but that's the neat thing about Wiki. Glycophi is the company. What did Glycophi, just to see if you followed, what does Glycophi do? In what way? Yeah, they did it but what significance? They were a company based on glycosylation of proteins, true. In yeast, okay. So they're the one that said they were going to add that to that. So that's obviously a way to even make faster ones, was to go that route with that, okay. But that doesn't necessarily deal with this issue of humanization. Now, there's chimera M things. So what they could do is they could go back to our antibody, which I erased here. Okay, let's get rid of ascites. That's kind of gross. And think of it in terms of there's this so-called constant and then there's the variable domains. And so there was chimeras, for instance, where what they would do is that part of it would be the human antibody, the constant piece, and then this might be the variable one that came from the mouse. They were the first ones in that. And then, what happened is and the way that was developed comes back to our mouse. Okay. So, what's the problem with the mouse? What?
It's a mouse.
It's a mouse. Okay. So let's be more specific about its problem as being a mouse, because we still can't put humans, because that would be the other way to make humanized antibodies, right? Would be just, you could just grow tanks full of babies. Throw them in lighters. Okay. But that's PETA. Beyond PETA problems. Okay. So what's wrong? So more specifically, it's not the mouse's fault, okay, that it's a mouse, but there is something about it that we would like to change. DNA is kind of too. I love that, guys. Say it right. Okay. Be bold in your mistakes. Okay. It is the best way to learn. Okay. You learn. Let's face it. You take an exam, you get 100%. Do you learn anything from that? No. You just like, I'll get out of here. Okay. DNA is kind of like saying, let's make. What do you mean? Okay. Like, who said it? Is that you? You saying DNA under your breath? Okay. I take away all the good stuff that you so Prometheus, you drink something and you fall into DNA parts. And, okay, sorry, I'm playing. Give me a break. Size saw hand.
Post-translation.
Post-translation, by the way, this is a glycosylated protein. So there are sugars on this thing. So that's part of the reason why there's a problem. But there's something else. What?
Well, the markers on the DNA, or when you make the protein, the markers on that protein for this mouse, specific to that mouse, so it'll recognize its markers.
Okay. Still not going to show you my scar. Okay. But let's say, for example, that we wanted to change our mouse to make our mouse human in the sense of its antibody. What could we do? We just need to put a human spleen in it, right? I need to make, yeah, okay. So if you think about it, if you can, what they did is they developed, there's a whole bunch of immune deficient mouse out there. And you can look them up. I'm trying to remember them. They're like the Nog mouse, and there's a Nog mouse, and there's a couple of other mice that are out there. You can buy them, okay? And the original one was the naked mouse. It was this mouse that was so screwed up. It didn't have any hair on it. It was a little feeble-looking thing. Okay. Okay, we're back to the mouse world. Okay. This is an ideal opportunity if you think about it, though. If I have a severely immune-compromised mouse, okay, what does that open the opportunity for? Yeah, we can transplant stuff into it, right? So the whole reason you have rejection of transplanted cells is because the immune system sees them. So immune-compromised mouse is a perfect candidate for a transplant. Okay? And what do we want to transplant into it? It's the ability to make B cells. Okay? So if I can introduce into this thing, basically, human, I don't really even want to do that. I'll put in chimera cells in that. Or if you remember what I talked about, there was those variable domains and all that whole editing thing that kind of slaps together the permutations of 10 billion options that you can make of that. Well, I just need to put that into the mouse, okay? And so I end up with, in fact, making one that is now a mouse. But if I inject it with an antibody, make sure we get the mouse right. Okay. If I inject it with an antibody, its immune response is going to be human. In fact, that whole editing process of it expressing them, the B cells with 50,000 things there waiting for the right partner to walk by, okay, and so forth. And then when it commits, then it doesn't really commit, and it has to change the way it interacts with time, and then the ones that don't interact die by apoptosis, okay, and the other ones tend to, that whole thing happens. And so you can end up then with it coming up with and selecting for a making of a humanized antibody now in the mouse. And it has rearranged its cells, unfortunately for the mouse. Now we need to go get its DNA, right? Because the B cells in that whole selection process that found just the perfect, really good binding in that, and now it's a humanized antibody, it's done this whole little DNA shuffling and point mutations and that to get to that antibody that is a human form, and it actually does, and if I could take that sequence now and stick that into a cell line, even like CHO or something like that, it will now make a human antibody, humanized antibody. One of the companies that was doing this was Metarex, and there's a whole series. I think there's about 7 or 8 antibodies on the market right now that were developed through immune deficient humanized systems like this. And there's also a humanized, I'm not even sure why it was a humanized cow, but there was also another company that I'm pretty sure made a version of a human mouse, which here we go. So there's a humanized mouse. Was Corinne? Anyone know the significance? They're a beer company. Okay. Exactly. Everything in Japan, they do one thing. They always do like 100 other things, like Mitsubishi makes everything. So for some reason, Corinne had a biotech division that made the human mouse too. Questions? Yeah.
How does this wrap into EPO? I mean, is this like...
EPO is a glycosylated protein made from, so here. Okay, so I decided to throw this in here because antibodies are so important and I don't have time to do an entire case study on antibodies. Okay, this is like a glycosylated protein, and in fact most current glycosylated proteins that are being developed in CHO all happen to be antibodies. Okay, so that's the connection to that system. And the technologies were the same. They went through the process of molar bottles, E-class, mobilized cell lines. They didn't go that